Welcome back to the Westin Copley Hotel here in Boston's Back Bay for our afternoon session. We'll kick off with a panel looking at gene and cell therapy delivery technology, a key technological challenge in the evolution of the field. And we can think of no one better to moderate this than Dr. Natalie Artzi. Natalie is appointed at Brigham and Women's Hospital, and she leads a multidisciplinary lab that's developing smart material platforms. Natalie?
Thank you very much, Chris, for the introduction. And we really can't discuss the delivery of gene therapy without mentioning the success of the COVID-19 vaccine. Could you imagine a nanoparticle delivering mRNA that otherwise would degrade in the bloodstream and will not be able to penetrate cells, then able now to successfully vaccinate millions of people around the world? While the COVID-19 vaccine race really propelled mRNA to the forefront of biomedicine, it is really important to enlighten the public about the role of the non-viral delivery systems that were developed really in the past few decades, and the hidden heroes behind the development of those platforms that enable such transformation, and some of them are here with us today. So welcome to all our panelists, and everyone have your bios, so let's really dive right in and begin by all the panelists really describing the key attributes of your technology. How is it different than viral delivery systems and any other gene therapy approaches? And maybe we can begin with you, Matthew.
Yeah, great. Thanks, Natalie, and thanks for having me. Our system really, we were founded at Generation Bio around the idea of breaking free from viral gene therapy. Now, it's first important to say, as you pointed out, that there have been tremendous successes with using viruses for gene therapy. But they have significant limitations, too, associated with the capsid. And so unlike many companies, we didn't start with the technology and then decide where to apply it. We actually just started with an idea of, can we break free from the limitations of capsid? And if you can do that, you really open up a number of things in terms of democratizing gene therapy at the standpoint of scale of manufacturing, cost. You mentioned mRNA vaccines. We are building a technology that is very similar when you think about manufacturing and scale. It also is a technology that's free from the limitations of size constraints of viral gene therapy of 4.7 kb. So we can do a lot with an expanded real estate. So we can add larger enhancer promoters, we can go after larger genes, we can put in regulatory elements. And then finally, our technology is really aimed at addressing the issue of redosing. Many patients already have neutralizing antibodies to AAV gene therapy. We can treat those patients because they do not have antibodies to our lipid nanoparticle technology. We can redose should expression wane over time, and that's an emerging consideration for gene therapy. And finally, and I think most importantly to us, kind of at the heart of what we're doing, is we can get into pediatric patient populations because we have this ability to redose. And that's where disease matters most, particularly for rare genetic disease, getting in early when you can change the course of the patient's life. To do that, we really had to develop, in-license one technology called closed-ended DNA. It really allows us to manufacture a DNA construct and get into the nucleus. And then, to your point on non-viral delivery, we had to actually engineer and build a new lipid nanoparticle system specific for DNA delivery. And those are the two technologies that are unlocking non-viral gene therapy for the first time.
Thank you. Laura?
Yes, and thank you for the invitation to participate in the panel. So I'm here representing Intellia. We are a therapeutic gene editing company. So what we are delivering, what we're using is CRISPR in its CRISPR-Cas9 technology in its various forms. So at Intellia, what we've done is deliver a strong platform with gene editing tools as well as delivery solutions that we can mix and match so we have the right combination for the type of edit in the cell type for a particular disease. as a company, we characterize ourselves as a full spectrum genome editing company because we're using CRISPR as a drug, and we'll talk a bit more about that with how we deliver it. But we also use it as a tool to reprogram cells ex vivo. And here is to do edits in a sequential manner, again, being very careful how we do these edits with the delivery, so the final product will have the intrinsic potency and the desired critical quality attributes. But talking more about in vivo and following on some of the points that Matt has made, we are delivering CRISPR-Cas9 to the patient in vivo. So there is an infusion of the CRISPR-Cas machinery to elicit the edit in vivo. And NTLA 2001 is our first candidate, the very first CRISPR candidate for systemic delivery that is in clinical trials. We've leveraged also a lipid nanoparticle. I like LNPs a lot as a non-viral delivery system. They are, precedented as a really effective, safe manner for delivering nucleic acids to liver. The beauty is non-limiting immunogenicity, pre-existing or de novo, as Matt said, large cargo capacity. They can go to the liver through platform innovation. You could push LNPs to go to other tissues, and we have recently shared some data about bone marrow delivery. The other key important thing is that these LNPs, at least the ones that we've developed, they are rapidly eliminating. And we believe that that's really important for CRISPR-Cas9. We want all of the elements to be there in the target organ long enough to do the edit, but then the editing machinery, which is bacterial in origin, as well as all the elements of the delivery vehicle to be rapidly eliminating. And that's what we have accomplished with our platform. So maybe I'll stop here and we can share more details.
Thank you. Yeah, absolutely. So we heard about LNPs, and now we'll move into exosomes. So maybe Doug, we'll go next to you.
Thanks, Natalie. Very nice to be here as part of the panel. I think what we're doing at Codiak is actually slightly different. Gene delivery is only one aspect of the way we're thinking about using exosomes as a delivery vehicle. For those of you who aren't familiar with exosomes, they are thought of as almost a FedEx system that cells use in the body. They're naturally occurring lipid nanoparticles with a complex proteome. And that proteome is important because it engenders immune silence. It engenders the capacity for trafficking, for uptake, and for delivery of the payload once the exosome actually enters the cell. So our platform allows us, and we've created a toolkit now that allows us to purposely change the proteome of the exosome, either to deliver a signal to a target cell or alternatively to alter the tropism. We can also load a variety of different molecules into the lumen of the exosome. They're protected in there. And this includes nucleic acids, small molecules, a number of other drug candidates that we can deliver using this technology. So we're in the clinic now with 2 candidates, both in oncology. Both of these candidates are really the first of their kind. They're the first engineered exosomes to enter the clinic. Some of the prior studies with exosomes have been done with unmodified exosomes, so naturally occurring exosomes taking advantage of the naturally occurring payload. But our approach is actually much more purposeful in that we've now created that toolkit that allows us to purposely change tropism, purposely incorporate drug molecules on the surface or in the lumen. We can combinatorialize if we choose to, given the simplicity of the way the platform works. And probably most important, given the fact that this is a complex modality, to be sure, we've developed the manufacturing technologies now to allow us to scale this up and have successfully done that in as large as the 2,000 liter scale with fed batch manufacturing. So we can engineer them, we can change their tropism, we can deliver them into certain body compartments of our choosing, And ultimately, we can make enough of them to think about this as a bona fide platform technology now.
Incredible. Thank you. And Sonya?
Hey, I'm coming after Doug to talk also about exosomes. As he mentioned, they're extracellular vesicles, and they're sort of the body's natural drug delivery system for macromolecules. At Evox, we are also engineering our exosomes and enabling them to, through our Intene technology, load proteins or enzymes. We can also load mRNA. We are looking and evaluating for at the platform level, taking advantage of that immunosilent nature of exosomes in order to look at loading up AAV-based gene therapy in order to be able to potentially deliver that without having that immune response that naturally occurs with AAV-based gene therapies. We have also developed our targeting engineering so that we are looking through systemic delivery to reach specific targets. And we are also utilizing and understanding the biodistribution of exosomes, both from their natural tissue tropism, plus when we can have them display ligands in order to improve that targeting via different routes of administration in order to access a CNS, for example. It's an interesting technology. I think we are kind of targeting different therapeutic areas potentially, but similarly, the CMC has advanced well within our hands, and we're at the 2,000 liter fed batch overgrowth process in place with kind of quite interestingly, we can develop stable cell lines in order to produce these exosomes with consistent quality characteristics.
Thank you. So to all of the panelists, do you view yourself then as a company with a platform technology that utilizes business, clinical opportunities to identify the indication, or do you choose an unmet need and then devise a new material or delivery platform for that? How do you choose your first indication? What's your approach?
Well, I can start. So at Intellia, for our in vivo pipeline, so we wanted to check two boxes. We do have a platform approach, and so it's important that whatever indication you select is going to have a read through to the rest of that particular delivery system and editing strategy, but also very importantly that there is unmet medical need and that the effort, the clinical candidate is going to bring benefit to patients. So for us, for in vivo, our first indication was transthyretin amyloidosis. So this is a severe disease where there is a protein that's produced by the hepatocytes in the liver that is pathogenic. It deposits in heart and in nerves, leading to polyneuropathy and cardiomyopathy that, if it's left untreated, is lethal. Over the last few years, there are new therapies that have been able to slow the progression of the disease, but there is more benefit to be gotten. And we believe that with our clinical candidate, NTLA-2001, we can not only match, but hopefully surpass the extent of reduction in that pathogenic protein, bringing more benefit to patients. So, the positives on picking that indication was that there were a number of already validated aspects of that drug development strategy. So protein made in the liver, we have a delivery vehicle to liver. We know what editing strategy to apply. We know what percentage of TTR would be clinically efficacious, right? So all of those elements were in place, leaving as the unknown the CRISPR-Cas mechanism of gene knockout. So, from that point of view, it serves the two purposes. And the approach, the platform approach, is that once you solve the liver and you have that proof of feasibility and safety in a particular cell type by, an editing modality and delivery modality, then you could go to any other gene target in that cell type using the same editing modality and delivery modality. So you hold everything constant, except for the guide RNA that determines the genetic location to introduce it. So there are a lot of platform benefits that can be realized.
Thank you.
Just to maybe follow on that, very similarly, we have an approach focused on the liver to begin with hemophilia A. We think there's a huge unmet need there, and very simple, very clear biomarker readouts for efficacy. So that's kind of a consideration. And similarly, as Laura talked about with Intellia, when you solve the kind of delivery system and construct for accessing non-viral gene therapy, we can quickly follow on in additional indications in the liver. One of the things we're most excited about is moving beyond just rare monogenic disease. And so we presented data at ASGCT last week where we're expressing antibodies from the liver for sustained applications in, for instance, antiviral indications. So the example we used was an anti-spike antibody in our collaboration with Vir. Now you can really take the power of this technology in terms of manufacturing scale and go into much more prevalent indications. And so that's the way we've kind of thought about starting with something that can get clear proof of concept, but then moving into more prevalent indications as we go.
Thank you.
I'll jump in if that's all right.
Yes, please.
I think we took the tack of, looking back historically at the starting point that some of the RNA therapeutics took. And in the early days, there was a lot of enthusiasm for antisense and siRNA, and really not a great understanding of what the possible was in terms of cell types that could be accessed by those constructs and where you could legitimately target pathways in those cells. So for a number of years, there was a lack of understanding about the real target cell spectrum that was accessible with that technology. And that led to, some of the work, particularly at Alnylam with the GalNAc conjugates, allowing for very specific hepatocyte delivery. Mm-hmm. And ultimately a different approach at Ionis, which was a chemistry-based approach, tweaked the molecule itself to alter the biodistribution. So understanding what your target cell spectrum is always a good starting point. Yeah. And I think for us, we certainly took that tack with the candidates that we've chosen to move forward, particularly true with STING, which is an engineered exosome that gives us very targeted delivery into antigen-presenting cells in the tumor microenvironment. And we didn't choose that target randomly. It's known that natural immune surveillance actually involves exosomes released from tumor cells being picked up by local antigen-presenting cells in the tumor microenvironment and stimulating the STING pathway. Right. So we essentially souped up what is a naturally occurring system in the tumor microenvironment to really try to start a productive immune response in the tumor. So we took nature as our guide, if you will, in terms of pointing us in the direction of where that system works naturally and really trying to modify it and enhance it and give it greater potency and specificity.
Wonderful. Sonya, would you like to add anything?
Yeah, so just our first internal pipeline program kind of takes advantage as well of our cell source's natural tropism within the liver delivery to hepatocytes as opposed to other cell types, and we've selected a urea cycle indication, ASA, and looking to deliver argininosuccinate lyase, which is the missing gene. What's interesting also about this particular indication, as well as having unmet need that we think we can address, as there's only dietary protein restriction and using nitrogen scavengers to address the complications of having high ammonia levels. We also know that we can get to extrahepatic tissues with our exosomes, and this particular component or enzyme of the urea cycle is also important to the nitric oxide pathway. And indeed, there are probably other tissue targets in the CNS that are important to deliver ASL to. So we thought it kind of met the kind of unique properties of our engineered exosomes, as well as being an area where there's kind of unmet need, and it's a difficult target for other approaches for enzyme replacement.
So absolutely, as we think about gene therapy and drug delivery as a whole, we would like to maximize the therapeutic index. And you alluded to this. We're using different strategies like administration mode and natural tropism of materials, and maybe even targeting ligands to drive our particles and delivery systems into a particular organ and cell. But how do you see that working when you think about combination therapy, where you may have different target cells? And Doug, you mentioned immune cells. Sometimes we want to target cancer cells with the chemotherapeutic drugs, for example, that are cytotoxic, but also we want the STING agonist to be uptaken by antigen-presenting cells. So how can a delivery system be used to target both?
I think it's understanding the cadence of the immune response is obviously an important part of choosing the strategy there. I think the advantage we also have here is that we can display exosomes, or we can display on exosomes a number of different ligands that would target different cell types. So a single therapeutic entity, the exosome, the engineered exosome, can actually display multiple ligands. We can incorporate things into the lumen of the exosome to
I think we lost you. Doug, you came in and out.
Do you want me to jump in? And oh, there he is. We have him back.
Doug, we lost you the last 30 seconds.
I'm sorry, did you lose the sound here?
Yes.
Sorry. It says that my internet connection is unstable, so I'll try to be more stable than my internet connection. Again, we have the ability with the toolkit we've created to display multiple ligands simultaneously quite readily. So within a microenvironment, you can engage targets on multiple cell types, multiple distinct cell types by virtue of that combinatorializing using the platform that's been created. So very straightforward approach.
Thank you.
Yeah, I would say at Generation we've taken a similar approach, but with lipid nanoparticles. So one of the things, necessity is the mother of invention. For delivery of DNA cargo, we recognized that we could not rely on lipid nanoparticles, kind of natural tropism, which is to hepatocytes via lipoproteins, but also to macrophages and immune cells. And coming back to STING, DNA delivered to immune cells is not a good thing if you want to increase your systemic therapeutic index for hepatic disease. So we actually took the tack of kind of engineering out the natural tropism to all the cells, basically, and then re-engineering through targeting ligands back to a specific cell type. And we started with hepatocytes because, as we've heard, GalNAc is a very nice, well-validated ligand with a receptor that's abundant in the liver. And that worked. We were able to expand our therapeutic index significantly. But an area we can take that further is if we swap that ligand for a different ligand, we think we can go after other cells. And we've done that in the retina. We've seen very good tolerability with lipid nanoparticles in the retina, which has been difficult to achieve with traditional lipid nanoparticles. And I think that flexibility of being able to control which ligands you put on our LNP and which cell types we get into will have that opportunity in the future to go after multiple cell types if we choose to do that.
Thank you. So, yeah, please, Laura.
No, I was going to say to add to Matt, right? So in addition of understanding the surface properties of LNPs and chemical modifications and targeting ligands is the ability of using different routes of administration, right? That can go systemically as well as local. And, that gives a lot of flexibility on choosing, therapeutic applications.
Absolutely, and in terms of patient compliance and ease of administration and redosing, that becomes also very important to consider what route we're choosing. Sonya, did you want to add anything?
Yeah, I mean, I think I think one of the advantages of exosomes is the fact that they are immunosilent naturally. And so in addition to trying to target through, decorating with ligands, we can also take advantage of that to deliver immunogenic cargo that's intraluminal, such as, siRNA, and therefore, would expect to be able to get, kind of better tissue uptake as well as an increased therapeutic index, whether that's through a local administration, such as, targeting in the retina or, or intrathecal. And I think it probably gives us a much better opportunity to get efficacious dosing without having the the safety issues that you see with response to highly immunogenic compounds.
Absolutely. So I think with the recent experience with the COVID-19 vaccine and everything that we've learned, we really are at an inflection point with gene therapy. The opportunities are vast. We heard about different delivery systems, different cargoes that can treat a range of diseases. I really am looking forward to seeing the impact of everything that we've learned, also in terms of capabilities of scale-up manufacturing, interactions with regulators. We've seen sometimes discussions and approvals overnight. So this is, I think, an extremely wonderful time for everyone that works in this area to really partner and work collaboratively to bring new therapies to the clinic. You may say I'm a dreamer, but clearly I'm not the only one. So thank you so much for all your efforts and for your insights today. And looking forward to following what you're doing and collaborating in the future. Thank you.
Thank you.
Thanks, Natalie.
Natalie.